David who?
David's right here.
Oh, okay. No, not that David. David
Brill?
Yes, it's David Bartel.
Bartel.
Here.
Oh, okay.
Present.
So, great. So we have our full thesis committee here with Emery Brown, myself, Lou Kunkel, and Dave Bartel. So we have a quorum, and we have an enthusiastic, hold the applause, like a political debate. No, you can fall short, you can equal time. But, so I first ran into Ali approximately 7 to 8 years ago. She camped outside my office, and it's been uphill since. And without further ado, Ali, why don't you tell us about what you've been doing the last few years?
Congratulations.
Okay, I'm very nervous. Hopefully it won't be too embarrassing. I want to tell you about my work on small RNA and A2I editing in autism spectrum disorders, which are a set of neurodevelopmental conditions characterized by deficits in social and communication abilities and an increase in repetitive and stereotypical behaviors. And behind this definition of a spectrum disorders lies a very broad range of disease manifestation, ranging from slightly awkward MIT classmate to really debilitating impairments. And this is a very big problem today because it's estimated that 1 in every 88 children in America is diagnosed with ASD, or autism spectrum disorders. And 85% of those are boys. So the plan for today is to first talk a bit about autism genetics, and I'll slow down, and then about gene-environment interactions of the synapse, and tell you a bit about our study that found that A2I editing might play a role in autism, and about a second study that looked into small RNA that are thought to regulate editing. Finally, what does it all mean, and where are we going from here? Ready? Sit. Go. Okay, although refrigerator moms were first blamed for causing autism, and then the MMR vaccines, today we know that autism has a substantial genetic component. And here you see the concordance rates for autism, which is the chance for one twin to have autism if the other one is diagnosed with autism. And here is the concordance for monozygotic twins. On average, it's 85%. For dizygotic twins, the average is 25%, and that makes autism one of the most heritable complex disorders. By complex, we mean that no single gene causes autism, but rather multiple genes responding to a set of environmental exposures. And tremendous progress has been made in discovering the identity of these genes. Today, we think that we know what causes autism in 20% of the kids. So the specific cause of the majority of cases remains unknown, but we have learned something, and that it is characterized by extreme locus heterogeneity. So what is this extreme locus heterogeneity? Here we have the human genome on the left, 24 chromosomes, and replicated linkage findings in autism are in green. Recurrent copy number variations are in blue. Genes causing symptoms in which ASD is a common symptom are in red. And non-syndromic genes implicated in ASD with high confidence are in black. So as you can see, on almost every single human chromosome, there are several regions implicated in autism. And that means that there are different causes for autism in different families. And therefore, they must converge at some higher level to give rise to a common phenotype that affects more than 1% of the population. And one such convergence is on synaptic formation and homeostasis. And many genes implicated each in a few families share a common theme of regulating synaptic formation and homeostasis. Here, as an example, is the glutamatergic synapse, and many genes in here were implicated in autism. So what we've just discussed is that autism is a complex genetic disorder. The cause of most cases remains unknown, but what we do know is that it's characterized by extreme heterogeneity on both the clinical and genetic levels. And one common theme that emerged is that many of the genes that cause autism are synaptic. And what we want to understand is what are the interactions between these genes and the environment that underlie this synaptic dysfunction and lead to ASD. And the human brain develops in response to environmental stimuli. Synapses are formed and destroyed in response to experience. This occurs throughout life, but there is an explosion of synaptogenesis during critical periods of brain development. And once a synapse forms, it remains plastic and changes in its properties in response to experience. So what is the molecular basis of this? Long-term memory is stored at the DNA level by chemically modifying the DNA or the proteins that pack it. And short-term memory is stored at the RNA level. Each neuron has thousands of autonomous synapses, and each synapse stores its own RNA pools and synthesizes proteins in response to its own dynamic microenvironment. And the ability to manipulate these local RNA pools using a variety of mechanisms provides an incredibly efficient means to fine-tune each independent synapse, which in combination with many other independent synapses and the central command of the nucleus provides an efficient way to integrate many levels of information and build complex models that are necessary for human cognition. And one of these regulatory mechanisms acting on synaptic RNA is A2I RNA editing. What is A2I RNA editing? It's a site-specific deamination of adenosine to inosine. It occurs mostly in the brain, it's neurodevelopmentally regulated, and it's carried out by enzymes that are called adenosine deaminase acting on RNA, or ADARs. Experiments in animals show that if you engineer their editing levels, you get altered complex behavior. And if you subject the animals to specific environmental exposures, they also show editing alterations. And therefore, it was hypothesized that A2I editing could be one of the molecular mechanisms that link between environmental inputs and behavioral outputs. On the molecular level, because inosine base pairs with cytidine, it's recognized by the cellular machinery as guanosine, and the impact of A2I editing is just like an A2G mutation on several transcripts. So if it occurs in coding regions of proteins, it can change the amino acid sequence and change their function. And this is one example of how A2I editing can change the function of synaptic genes, and specifically synaptic receptors that are very important in synaptic plasticity. These are 5 ionotropic glutamate receptors, and the 3 receptors to the left here undergo editing in the middle of a transmembrane domain that changes a neutral glutamine to a positively charged arginine, and this dramatically reduces their calcium permeability because now there's a positively charged amino acid in the middle of the channel. Another example is in these 3 AMPA receptors to the right, where an R2G change, arginine to glycine, leads to faster desensitization and recovery of these receptors. Altogether, because editing can affect between 0 and 100% of the molecules, it provides a very efficient means for regulating synaptic strength and duration, which is the basis of synaptic plasticity. So I really wanted to test the role of A2I editing in autism, not only because it's a relevant mechanism for gene-environment interactions that underlie a common etiology of synaptic dysfunction in autism, but also because there is increased editing in the human lineage. And there are autistic-like symptoms in animals when you engineer their editing levels. And finally, the Fragile X fly has significant editing alterations because the Fragile X mental retardation protein directly binds the editing enzyme in the fly. And so now I'm going to tell you how we set up a system to test the involvement of A2I editing in autism and what we found. So for an initial examination of A2I editing in autism, we decided to focus on post-mortem cerebellum because that is one brain region that was consistently implicated in autism using both post-mortem and imaging data. And luckily, around that time, brain banks in the country started collecting high-quality tissue from individuals with autism, and we were able to obtain 30 samples from individuals with ASD and neurotypical individuals matched by the criteria shown below. Out of those, 25 had high-quality RNA, and they were included in all of our previous studies. And for an initial examination, we wanted it to be very focused and with very clear functional implications. So we decided to look only at 10 synaptic genes that are known to undergo neurodevelopmentally regulated editing with very clear implications to synaptic transmission. And our approach was targeted multiplexed ultra-deep sequencing of cDNA, which was coupled to genomic DNA genotyping, and an A2G mixture on cDNA that was mapped to a homozygous A was indicative of RNA editing. And this is how the experimental design looked like. From each tissue, we extracted RNA, and then we synthesized cDNA, and we selected targets for sequencing using 2 rounds of PCR. The first one was gene-specific, and the second one was sample-specific, and added a barcode to each amplicon based on an anchor that was part of the primers of the first PCR. Each PCR was purified, and then we quantitated the products and pooled them in equimolar amounts for sequencing as 2 pools on 2 sides of a 454 picotiter plate. And in parallel, we also extracted genomic DNA and genotyped it using sequenom from the exact same tissue section. And I've had the great privilege of doing all this lab work with Kayla and Jill. And we got more than 1,000x coverage from the 454 sequencing run, which enabled a high confidence data set of human RNA editing. And the relatively long 454 reads of about 200 base pairs let us examine the context of editing. For data analysis, our goal was to find the most likely editing level given the sequence data and provide a confidence estimate for that measurement. And so the way it looked like was for each individual at each site, we had n total reads, and out of those, k were edited. So to rephrase our goal, it is to find the density function of the editing probability p given n and k. And thanks to Professor Brown, we used the beta binomial framework to update a prior belief of the editing probability with observed binomially distributed data, and we got a posterior beta editing distribution that was based on both the total number of reads for each individual at each site and the fraction of those that were edited. And this is how it looks like visually. These are the posterior editing densities for 25 individuals at one site, BRIC2QR. And each individual is depicted by 1 posterior beta density. And the peak of the density is the point estimate for the editing, and the width is the confidence. The taller it is and the narrower it is, the more confident we are in the measurement. So for this one site, BRIC2QR, we have a very wide range of editing levels, going from 30 to 90%. And I didn't tell you that individuals with ASD in this color coding scheme are in orange, and neurotypical individuals are in blue, and throughout all of the slides, and this will be the color coding scheme. And we can later group individuals by their affected status and make comparisons between individuals with ASD as a whole and neurotypical controls, and this is shown here at the bottom. This is one site. We did the same thing for 25 different sites. Some of them were known to undergo neurodevelopmentally regulated editing, and others were identified by more than 30,000 cDNA reads mapped to a homozygous A and with an A to G mixture on cDNA. And we wanted to validate our approach. And for that, we used an independent method that uses a slightly different targeting approach, which is using padlock probes, and a different sequencing platform, which is Illumina sequencing. And this was done in collaboration with Billy Lee at Stanford University. So we measured the editing levels of all individuals in 6 sites using the 2 methods, and we found very high correlations between the 2 editing detection methods. So we think our method seems to work, and what do we find with it? We find a very high dynamic range of editing with ASD at the extreme. Here on the left, you can see the mean editing fractions and standard deviations among all individuals at all sites, and you can see that it goes from below detection limits, which is 2.5% editing, all the way up to nearly fully edited at 99% editing. And this dynamic range is consistent with a role for editing in fine-tuning synaptic function in response to individual environments. On the right, we have the individual deviation from the mean, and you can see that for almost all the sites, actually 20 out of 25, the extremes are orange triangles, which are individuals with ASD. And in fact, being at least 2 standard deviations away from the mean is highly indicative of ASD with a positive predictive value of about 80%. And this is the take-home slide from our study. It shows the differences in the landscapes of synaptic editing in individuals with ASD in orange, neurotypical individuals in blue, editing sites on the horizontal axis. Individuals are coming out of the wall, and each matched pair is overlaid on one another. And so you can see that across almost all the editing sites and different individuals, and the individuals with ASD tend to have either too much or too little editing as compared to the mean, and specifically to their matched control. And here on the left is a histogram that summarizes the deviations from the mean. And you can see that the extremes are specific for ASD, and only individuals with ASD were found more than 3 standard deviations away from the mean editing level. So next, these are quite substantial differences, and we next examined what could cause these differences. And one potential cause for them is altered function of the editing enzyme, ARB1. ARB1 is the enzyme that regulates editing at most of the sites that we measured in the study. And it's known to undergo neurodevelopmentally regulated alternative splicing that removes this exon shown here that holds the double-stranded RNA binding domains of the enzyme and makes it dysfunctional. And it's untranslated and therefore dysfunctional. And so we use semi-quantitative RT-PCR to measure the relative frequency of the dysfunctional isoform using primers on the exons that flank this alternatively used exon. If you get a 122 base pair product, that is indicative of the dysfunctional isoform, and we could measure the relative frequency of this out of the entire PCR product to find that individuals with ASD use the dysfunctional isoform about twice as much as neurotypical controls. And the usage of the dysfunctional isoform was correlated with overall editing levels. And because this was an important finding, we validated it using an independent method in the same cohort and the same method in an independent cohort. And with the help of FEDEC here, we used qPCR in the same 25 samples, and we used an independent cohort of brain samples with the semi-quantitative PCR, also RNA-seq on a subset of the cohort that we measured editing for. And all of these methods agreed that individuals with ASD use the dysfunctional ARB1 isoform much more commonly than neurotypical controls. We next looked at editing patterns of specific candidate genes for autism. And here is the serotonin receptor, HTR2C. It's a very important candidate gene for autism, and we're going to describe it in more detail later. For now, you should know that it undergoes editing in 5 sites that are found in its G protein binding domain. This is a G protein coupled receptor. And combinatorial editing in these 5 sites may lead to up to 24 different protein isoforms that have a substantially different ability to bind G proteins. And when we compare the relative frequency of these isoforms in ASD versus controls, we found that they're significantly different, but we have to be very careful about this finding because the variance in individuals with ASD is much greater than in controls. So this finding has got to be replicated in a larger cohort. We also looked at the ionotropic glutamate receptors that I've shown you before and compared their editing levels between ASD and controls. GRIC2, shown here, has 3 editing sites in combination, and they can lead up to 8 different isoforms, and we found a differential usage of these isoforms. And we also found significant differences in the levels of editing of the R2G site in GRIA4. So next, because this was the largest study of human A2I RNA editing, we wanted to test some hypotheses that were previously made, but it was not possible to test them before. And one of them was about the relationships between editing and splicing. Another that I'm going to discuss was between editing sites in cis, and the last relationship that we examined was between editing sites in trans. And the 3 AMPA receptors, GRIA2, GRIA3, and GRIA4, use alternative splicing to control the receptor desensitization kinetics by including either the flip or the flop exon. And it was thought that the usage of this flip-flop cassette is related to editing in this site right here, which is found immediately upstream to these 2 exons. And because we had a lot of data, we were able to count the number of co-occurrences of editing at this site and usage of flip or flop exon, and we found that the edited version of exon 13 gets preferentially spliced to the flop isoform, flop exon, and the unedited version is significantly associated with usage of the flip isoform, and you can see the odds ratios below. Another relationship that we examined is correlation between editing and intron retention in filament A. And when we tried to amplify across the Q2R editing site here in FLNA, we found an unexpected band of 720 base pairs in every single sample. When we sequenced it, we found that it is a result of intron retention. And here you see the alignment of the Sanger sequencing grid to the genome and to all the known isoforms of FLNA which lack this intron. We found that the Q2R editing is significantly correlated with usage of this intron, with retention of this intron, but there is no one-to-one relationship. What we did find is that every single intron that was retained included a novel A2I editing site. And some of the transcript also had another A to G mixture on cDNA, which when we genotyped the genomic DNA, we found that it's in fact a genomic SNP, and it actually has a DB SNP ID shown here. Another relationship that we looked at was between 3 editing sites that are 4 base pairs from one another on GRIA2, an AMPA receptor. Here we have GRIA2QR editing, which is almost always edited in almost all transcripts. Not immediately, 4 base pairs upstream from it is a site that is barely ever edited, and 4 base pairs downstream from it is a site that gets edited variably between 0 to 20% in different individuals. And because we had a lot of data, we could confidently say that they are independently edited, either by different complexes, or at different times in development, or in different cells. We could also examine relationships among editing sites by calculating the correlation coefficient of their editing levels across all individuals, and then by clustering the data. And here you see the tightest clusters that we found that are suggestive of co-regulation in cis and in trans. Summary of this study is that we found that 30% of the individuals with ASD were the extremes of synaptic editing distributions. We identified differential editing of ASD candidate genes, and found differential splicing of the editing enzyme ARB1, which was correlated with editing levels. And we identified relationships between splicing and editing, and relationships between editing sites in cis and in trans. Next, I want to tell you about our follow-up study that looked at how small RNA that originate from a genomic locus implicated in autism could potentially regulate the editing of the serotonin receptor, HTR2C. And HTR2C is an important functional and positional autism candidate because serum hyperserotonemia is the most consistent biomarker in ASD, and many genes in the serotonergic pathway were genetically implicated in autism. Also, biochemical pathways of serotonergic transmission in the brain are differentially used in individuals with ASD as compared to controls. And HTR2C is the target of the 2 drugs approved to treat autistic symptoms. It is an X-linked gene, and autism is a male-dominant disorder. And finally, editing levels were shown to be different in a mouse model of autism. And as you remember from the previous study, we measured the editing levels in these 5 sites. The serotonin receptor, we found differential usage of editing-mediated isoforms and said that it has to be replicated in a larger cohort because of the increased variance in autism. And it is thought that this editing at these sites is regulated by this small RNA shown here, which is a CD box small nucleolar RNA, or SNORD115, because it has an 18 nucleotide long stretch of complementarity around the edited region. And the CD box designation is based on 2 sequence motifs that are found in all of these small RNAs, a C box and a D box. And these SNORDs originate from one of the most commonly altered loci in autism, the 15q11 locus, which is an imprinted locus in the human genome. and they are found right here. They're expressed from the paternal allele, and if you have mutations that lead to lack of expression from the paternal allele, you get Prader-Willi syndrome, and if you have mutations that lead to loss of function or no expression of maternal genes, you get Angelman syndrome. And for males with Prader-Willi syndrome, there is a 40% chance of having a comorbid autism. For females, there is a 5% chance for having autism. And this 8-fold male to female ratio is almost twice as much as the general population male to female ratio in autism. And the SNORD115 genes, the SNORD115 is actually a cluster of many highly similar genes, 48 of them, and which are all thought to regulate the serotonin receptor. And immediately upstream to it is another small RNA cluster, SNORD116. It includes 30 genes, and it's thought to be the critical region for Prader-Willi syndrome. And here are some recent findings of contradictory relationships between SNORD115 expression and HTR2C editing. Although, remember, the sequence complementarity is suggestive of some RNA-level relationships, and these, you'll see, are not consistent. And in mouse, there were several studies. One of these showed that if you transfect a generic mouse SNORD115, and you get reduced HTR2C editing in neuronal cell culture. But in vivo, in a transgenic mouse that had 2 copies of the SNORD115 cluster, there was an increase in HTR2C editing. In men, there were 2 small-scale studies. One looked in 4 Prader-Willi individuals that have a SNORD115 deletion and compared them to 2 neurotypical individuals and found that the deletion is associated with increased editing of HTR2C. But another study using 2 different individuals with Prader-Willi syndrome as compared to 3 neurotypical individuals showed that a SNORD115 deletion has no effect or is not associated with any changes to serotonin receptor editing. So we wanted to know what exactly is going on over here and what are the physiological relationships between this small RNA and HTR2C editing, and what is the overall contribution of SNORD115 and SNORD116 to idiopathic autism. So for that, we used deep targeted sequencing of these SNORDs from the same samples that we profiled editing on, and we compared editing levels from the previous study with that expression. And there are 2 main differences between this and any generic small RNA sequencing study. First, it's a targeted study, meaning that only genes with a specific sequence are selected for sequencing. And second, it enables direct inter-individual comparisons by using a normalization spiking. So let me just go over these 2 points to show you how they worked out. The selection was based on multiple sequence alignment shown here that identified conserved regions in color at both ends of all of these SNORD115 and SNORD116 genes. So on these conserved sequences, we designed degenerate PCR primers that amplified all of these genes in one reaction. To enable direct comparisons between individuals, we spiked the reverse transcription reactions with a synthetic SNORD. We made it to have similar length and GC content as the endogenous SNORDs, but it lacked any match to the human genome, so we could confidently quantitate it. And then we amplified it with the endogenous SNORDs, purified, barcoded, and sequenced the PCR products as before using 454 sequencing. For data analysis, we had quite a challenge because of the high redundancy between highly paralogous genes. And we couldn't use any off-the-shelf quantitation method, and in order to identify the most confident one, we used simulations based on an empirically derived 454 error model. And we tested different pre-processing and genome alignment strategies, and also different considerations for expression quantification. And these let us identify which are the genes that we can, no matter what, we will never be able to distinguish between. And we grouped those in families, and then iteratively optimize the method, finding the minimum number of families that would be confidently detected. And we found 55 such families made of 78 different SNORD genes. And once we defined the optimal quantitation method, we generated 2 additional simulated data sets to test it, and we found that it was highly accurate. And so now we can actually use it and see what we find. And here you see the subcluster cerebellar expression of SNORD115 and SNORD116 families. On the y-axis is the total number of reads relative to the spike in. On the x-axis is the genomic coordinates of each of these SNORD families. This is the SNORD116 cluster. This is the SNORD115 cluster. You can see that most SNORD families were indeed detected, and there are several dominant ones within each cluster. And so now we can finally examine the relationships to HTR2C editing. And first we wanted to be able to compare our results to previous studies. So we examined the overall SNORD115 expression level relative to the spike in here on the x-axis, and the relationships with the fraction of HTR2C transcripts edited at a specific site. This is site A. And we found that most samples have both a low editing level and a low overall expression level, so they're not very informative to identifying relationships. And altogether, there were no obvious relationships between the 2 variables. We did the same for all 5 sites, and no obvious relationships emerged between overall SNORD expression level, which is what was measured in the studies that I've described before, and single site editing at each of the 5 sites. But what we were really interested in is identifying the relationships between specific SNORD families and specific isoforms that are as a result of combinatorial editing, because this is how the relationships actually work in the cell. It's not a single site level thing. And so here on the left you can see the alignment of the SNORD families to the HTR2C transcript. The edited region is shown here, and not all of the SNORD families actually align to the serotonin receptor. And we use those that actually align for multivariate linear regression to identify significant relationships between the combination of SNORD families and editing-mediated isoforms. We did find a significant relationship, but it likely does not have a very big contribution because the relative frequency of the isoforms that are thought to be linearly correlated to SNORDs is very, very low. And so we conclude that the regulation of HTR2C editing is very complex, and SNORD115 may play a non-dominant role in it. And then the next obvious thing is to compare the levels of expression of the SNORD families between autism and controls. We did not find any significant differences, but unsupervised analysis showed that there is a very strong internal structure to the data, and that was related to gender. So then we directly compared genders between, of course, males and females. Males are gray and females are purple, and as you can see here, there is a consistent 2-fold overexpression in almost all of the families across the region, both SNORD115 and SNORD116, and this could be important in delineating the contribution of this locus to autism, which is a male-dominant disorder. Next, we examine the relationships between overall SNORD115 expression on the x-axis and SNORD116 expression on the y-axis, and these are known to be independently transcribed. We find that they're very tightly correlated, which suggests that in human cerebellum they might be co-regulated. Finally, we're almost done. We use this data to identify putative, novel, previously uncharacterized CD box RNAs. And this was done by having multiple reads from multiple samples aligned to an uncharacterized location in the genome. And here you have 15 reads from 7 different samples, both ASD and controls, males and females, that all have a C box and a D box, which is the characteristics of CD box RNAs. And in order to be a functional RNA, they must fold into a stable secondary structure, which is shown here. And this specific set of reads aligned in antisense to the 3' UTR of myelin basic protein, which is brain expressed genes. And in fact, 13 out of 20 confidently detected putative SNORDs are cis-antisense to 3' UTRs of brain-expressed genes. So this could hint to their function. In summary, we were able to examine the physiological relationships between SNORD115 expression and HTR2C editing in humans, and we found nonlinear relationships between overall SNORD115 expression and single-site editing. We found weak linear relations between specific SNORD115 families and HTR2C isoforms. We performed direct inter-individual comparisons of subcluster SNORD115 and SNORD116 expression, and we found a consistent twofold overexpression in males. We found that overall SNORD115 and SNORD116 are tightly correlated, which is suggestive of co-regulation, and we detected a very small number of previously uncharacterized SNORDs, and 65% of those are cis-antisense to 3' UTRs of brain-expressed genes. So what does this mean? And what is the overall impact of this work and our future directions? Overall, we have an accurate comparative quantitation of small RNA and A2I editing in a human brain population. We found that synaptic A2I editing could play a role in ASD. We hypothesized that it could be one common compensatory mechanism to a very wide array of different synaptic insults acting on affected neurons in autism. But in order to establish the exact contribution, more genes and more brain regions and more individuals must be examined. And we are in the process of doing exactly that. Another future study is to look at the diagnostic potential of editing detection in the blood. And finally, another study that we should finish in the next few months is survey of microRNA editing, which are very important regulators of synaptogenesis and prime editing targets in the same samples, and we want to look at their potential contribution to autism. So, and I want to thank everyone, and everything you've seen and heard today was made possible by my two dads, Zach and Lou. and I really want to thank you for all these years of support and inspiration and amazing training and mentorship. Thanks. And I'd also like to thank my gurus, Professor Emery Brown and Professor David Bartel, for investing your time on my committee and your amazing contributions to my work. And to Uncle David, thanks for being a great mentor in the past few years. And many, 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 many thanks to all, really the best lab mates in the world for all your help throughout all these years. And to my close friends and family, I wouldn't have made it without you. I love you very much. Thanks, everyone.
To observe, so, even though you invite the Supremes, does anybody have any questions? Any questions? Any questions, lay or scientific, are good. I'll just try to warm up by asking you a question. You did some very nice work to show, to demonstrate, I think, very, very clearly that the autism brains were on the extremes of the distribution in A2I editing. But if you squint and forget statistics, it really looks, as you highlighted to us, that everybody's sort of the same, but then there's these outliers.
Mm-hmm.
So do you think that if you had many more, 100 such brains, which we might actually ask for a contribution right after this meeting, if you had 100 such brains, would it be a smooth function, or do you think it's really, these are outliers, or is there going to be a smooth distribution? In other words,
Yeah.
do you think these are big whacks, or we're going to see?
I think it will be a continuous distribution.
You'll think it'll be a continuous distribution?
Because of the dynamic range that we did see. Do you want to see it again?
No, I believe it. Any other questions?
Shosh, do you have a question?
Not yet.
Yes.
That was a great talk, Ali.
Thanks, Raj.
So you looked at the overdispersion of A-dye editing in ASD candidate genes, but if you look at non-candidate genes, would you also expect overdispersion of A-dye editing?
Very good question, and I didn't tell him to ask it. So we looked at a number of genes, and we find that those that are involved with synaptic function have an increased level of increased extremes and that are populated by individuals with ASD as compared to non-synaptic genes. And we should write that up soon. So we think that it's specific for synaptic genes, not all together, all RNA genes. And of course, not all individuals with autism. Yes, Matt.
SNORD 115 and 116, how many complementary regions with any of those autism?
And we are examining that because it's unclear what degree of complementarity would support the regulation. We have a few regions that we think that SNORD 116 might regulate alternative splicing because it's aligned to an intron in the vicinity of 2 exons, and we do examine that. We're working on validating it using other people's data.
So just a general question. So what's the current thinking as to why this is more prevalent in men or boys?
Well, some suggest that autism is an extreme male brain phenomenon where you know that a priori females tend to be much more social and communicative, and you guys are predisposed. I'm sorry, Anne-Marie. And of course, many have focused on the X chromosome. The X chromosome has many synaptic genes, and we can go back to the slide that shows that it has an enrichment of genes that are thought to be implicated in autism. And so it could be not only X-linked genes, but also autosomal regions that could be differentially regulated, such as this 15q region, between the sexes in the brain. And there are many genes in the brain that are differentially expressed between males and females that are on autosomes, not just X-linked genes. There's a very, very nice recent study in Nature, Kang, from 2011 that describes this.
Any other questions? Well, exceptionally, because of the mass turnout, I'm going to ask the thesis committee to indulge me and say, instead of getting everybody to move out of the room while we discuss her, let's, as a thesis committee, move out of the room, leave everybody here, and discuss.
Right. Alan, did you get another room?
No, but I saw that there are some things outside.
So I invite the thesis committee to join me.
Deliberate.
Very annoying.